we will consider about the next part of the lipid metabolism, that is about the phospholipid metabolism. Already we have considered last presentation about the triacylglycerol metabolism, the intermediate is phosphatidic acid. Now we'll consider about the next part. This phosphatidic acid is also involved in the synthesis of phospholipids. As we know, there are various different phospholipids. Already we have discussed about. We'll consider about this phospholipids in detail. So, as previously known, the synthesis of triacylglycerol is by distillation of three hydroxyl groups of the glycerol 3-phosphate destetrates to form the triacylglycerol molecule. And for the formation of this triacylglycerol molecule, there is an intermediate, what we know is as a phosphatidic acid, whereby one phosphate group is attached and two fatty acids are esterified. This is called as 1,2-diacylglycerol phosphate, that is nothing but phosphatidic acid, is the common intermediate for triacylglycerol synthesis as well as phospholipid synthesis. Now, already phosphate is there. Now there will be the addition of only additional nitrogenous group. There are different nitrogenous group like choline. Then we get this lecithin phospholipid. If ethanolamine is added, we get the cephalin phospholipid. If there is a addition of inositol, we get phosphatidyl inositol. Likewise, we get phosphatidyl serine and phosphatidyl threonine. Now, how exactly these different phospholipids are synthesized? We'll by step in detail. Now here you can see a complex metabolic pathway showing the different products of the phospholipids produced. So as we know earlier, we get first the 1,2-diacylglycerol phosphate. Now these are 1,2-diacylglycerol phosphate is obtained from two sources, either from dihydroxyacetone phosphate or from glycerol 3-phosphate. If we start from the dihydroxyacetone phosphate, it is first undergoes the dehydrogenation reaction to form glycerol 3-phosphate, and then subsequent reaction does happen. So finally we get the 1,2-diacylglycerol phosphate, which subsequently undergoes phosphate group removal and finally addition of one more fatty acid to form triacylglycerol in a usual way. And in the second way, if the choline, that is the nitrogenous base, is present, now this choline undergoes first phosphorylation activation by kinase reaction, and subsequently it binds with the CTP molecule, which is a nucleotide cytidine triphosphate, and here the transferase enzyme transfers this choline to form a CDP choline. Now this CDP choline becomes the choline group donor for this 1,2-diacylglycerol, and subsequently next transferase reaction we get the phosphatidyl choline, 1,2-diacylglycerol phosphocholine, that is the lecithin is synthesized. Now this lecithin in the similar way, instead of choline, if there is an ethanolamine, so ethanolamine also can get activated and forms CDP ethanolamine, and that can get added to this 1,2-diacylglycerol, and it will form the phosphatidyl ethanolamine, just like here shown here. So phosphatidyl ethanolamine will be synthesized. Now phosphatidyl ethanolamine has got one more liberty, so it can exchange with serine to form phosphatidyl serine, and ethanolamine can be released. So likewise, we get the first phosphatidyl choline, lecithin, then there will be formation of phosphatidyl ethanolamine, then there will be formation of phosphatidyl serine. So all these are the phospholipids of the plasma membrane at various sites. So predominantly phosphatidyl serine is generated during the synthesis of phospholipid bilayer of the plasma membrane. Now one more point here to be added. This phosphatidylserine can undergo decarboxylation and it can form the phosphatidylethanolamine. There is one more reaction to form the phosphatidylethanolamine. Now, on the other side, the next components, there are cardiolipins. These are also synthesized. Phosphatidylinositol, that is also synthesized. And how exactly these are synthesized? Now, in general, whenever the phospholipid synthesis does take place, whenever the glycolipid synthesis also does take place, commonly these metabolites undergo first basic reaction, that is the combining with the nucleotide to form nucleotide phosphate. So CDP choline, CDP diacylglycerol or CDP ethanolamine, CDP glucose, galactose, etc. So either the 1,2-diacylglycerol or the additional compounds, they can get combined with the CDP and they becomes the donor for the next compound. And in both the way, there will be synthesis of different phospholipids, even also the glycolipids. So in the subsequent reaction, 1,2-diacylglycerol phosphate, it is going to form 1,2-CDP diacylglycerol by combining with the CTP again, just like the choline on this side. And this CDP diacylglycerol will combine with glycerol 3-phosphate to form cardiolipin. We'll see in the next present slide. Or it can also combine with inositol. It can attract one inositol molecule and it forms phosphatidylinositol. Now this phosphatidylinositol is a very important plasma membrane phospholipid because it is involved in various activities like second messenger, signal transduction, and so on. So this phosphatidylinositol undergoes subsequently kinase reaction and it gets phosphorylated in response to the action of neurotransmitters or hormones. So phosphatidylinositol has ability to phosphorylate additional hydroxyl groups and it forms phosphatidylinositol 4,5-bisphosphate. Now this phosphorylation is also required. It is one part of the reactions whereby whenever the hormones bind to the receptors on the plasma membrane, they can generate the cascade of reactions, and few of these are this, and then there will be again the lysis. Degradation of these phospholipids also does takes place, and then we get the products which becomes the second messengers. That is also coming in the subsequent slide. So here at present we will just remember that CDP diacylglycerol combines with inositol to form inositol phosphatidylinositol and it can get phosphorylated to phosphatidylinositol 4,5-bisphosphate. Now the next, again about the phosphatidylcholine, we will consider now this phosphatidylcholine has got two ways of the synthesis. First way of synthesis, we have seen that 1,2-diacylglycerol takes up the CDP choline. Now, where the CDP choline comes from? The CDP choline is a choline that is already present within the cells because of the hydrolysis of existing phospholipids. So, in most of the conditions, so whenever the phospholipid synthesis takes place, it prefers first the utilization of existing choline or ethanolamine or other amino acids to form the phospholipids. But this choline moiety can also be generated de novo way, means completely new choline molecules can be synthesized after their incorporation into phosphatidyl groups, phosphatidylate. So we know that phosphatidylserine by exchange from decarboxylation reaction from phosphatidylethanol or by exchange with the serine. Now this phosphatidylethanolamine, what does it undergoes transmethylation reactions by with the help of S-adenosyl methionine and transferase enzyme, but it has to undergo 3-methyl methylation reactions, because to ethanolamine to get formed into choline, three methyl molecules will be required, and three methyl groups will be donated by methionine in the form of S-adenosyl methionine, and there will be subsequent formation of S-adenosyl homocysteine. Again, the three molecules will be generated of S-adenosyl homocysteine, and then there will be the formation of phosphatidylcholine choline. But economically for the cell, this is little bit expensive because we will require here lots of other ingredients like S-adenosyl methionine. Methionine will be the requirement. Folic acid and vitamin B12 will also be utilized in this reaction. But this is also the one of the way of formation of lecithin. Now we next consider about the cardiolipin. It is a CDP diacylglycerol will donate this diacylglycerol to glycerol 2 phosphate to making form phosphatidylglycerol phosphate. So diacylglycerol phosphate group and there will be a glycerol phosphate on the side. They combine to form phosphatidylglycerol phosphate. Subsequently, it is going to form the phosphatidylglycerol phosphate will be released, and it will take one more diacylglycerol and it will form the diphosphatidylglycerol. That is nothing but the cardiolipin. It has got bulky structure. It is present in the plasma membranes of the mitochondria, and it is involved in the process of programmed cell that called as apoptosis. In addition to this, other phospholipids are also involved in the process of apoptosis. Now, regarding the phosphatidylinositol, it acts as a second messenger. It acts as a second messenger. In a short time. Before that, we consider just now, once these phospholipids are synthesized, they undergo constant modifications. Post synthesis, they undergo constant modifications. Being a component of plasma membranes, whenever these phospholipids are required, so from the existing uh, diacylglycerol, monosacglycerol, which will be the part of plasma membrane fluid model, and they will be selected by the specific enzymes, and they will be either phosphorylated or choline will be different groups will be added, and they will be synthesized into different types of the phospholipids. So actually the phenomenon does takes place likewise at the site of the plasma membrane. On the other hand, as and when required or because of the some activities or regulatory forces, so these get catabolized at different sites. So for the degradation of phospholipids, phospholipases are the enzymes, and they are of different types. The first one is the phospholipase A1, which releases the fatty acid at C1 position of the glycerol, and it forms the lysophospholipid and free fatty acid. Subsequently, phospholipase B, which releases the fatty acid at C2 position, and remaining remaining part of the glycerol with phosphate and nitrogenous base. So there is one more enzyme known as the phospholipase A2. This phospholipase A2 is little bit very specific because it releases the fatty acid at C2 position, releasing the lysophospholipid and free fatty acid. Now, the significance of this enzyme is that first, it is found in the pancreatic juice, and it is present in different cells also, because most of the phosphatidylinositol at C2 position, and most very commonly phospholipids at C2 position contains the unsaturated fatty acids. These unsaturated may be polyunsaturated, long chain, etc., whatever it may be. And predominantly in phosphatidylinositol, this fatty acid is nothing but the arachidonic acid, and arachidonic acid is required for prostaglandin synthesis. So as and when this prostaglandin synthesis is required, cyclooxygenase or lipoxygenase enzymes, if they are present in the vicinity, so they are made available with the arachidonic acid by the action of phospholipase A2, and lysophospholipid will be released. So that is about the phospholipase A2. Now it will be activity of this phospholipase A2 can be inhibited by the glucocorticoids or cortisol. So, so it becomes the important finding that it can be utilized for various clinical purposes. Now, next is the phospholipase C. It is found in the liver and it is also found in the toxins of the Clostridia and different bacilli, and it will release this phosphate plus nitrogenous base, leaving behind the 1,2-diacylglycerol. And phospholipase D will release the nitrogenous base, keeping intact the phosphatidic acid. It is found in the plant tissues. Now, similarly, phospholipase C acts on the phosphatidic inositol 4,5 bisphosphate. We have already seen that CDP-diacylglycerol combines with inositol from inositol phosphatidyl inositol, and it gets subsequently phosphorylated to form phosphatidyl inositol 4,5-bisphosphate. Now, this phosphatidyl inositol 4,5-bisphosphate will be acted upon by phospholipase C. It will be activated because of the binding of carbons to the receptors, and it will release the diacylglycerol and inositol triphosphate, and these two molecules becomes the second messengers. They activate the different other enzymes and they exert the effect of that hormone within the cellular environment. So accordingly, this phosphatidyl inositol is very uh, important, and it undergoes the phosphorylation in response to the receptor binding on the plasma membrane, and it results in the, so this release results in the mobilization of intracellular calcium, activation of different protein kinases, and then subsequent cascade of the events exerting the hormonal effect at the metabolic level. Now, another point regarding the phosphatidyl inositol, these phosphatidyl inositol proteins can covalently bind with the proteins on the exterior of the plasma membrane via the carbohydrate bridge. So here are some enzymes like alkaline phosphatase, acetylcholine esterase, lipoprotein lipase. So uh, they can bind with this group and they can be transport laterally along the plasma membrane. So acetylcholine esterase is found in the postsynaptic membranes, and they can degrade acetylcholine. And again, it is found in the plasma membranes of the some protozoas. So it can anchor the protein on its structure, and it is hydrolyzed by this phospholipase C enzyme. That is about the phosphatidyl inositol. Now, the next part, there are other phospholipids which are known as the plasmalogens. Plasmalogens, and there is one more specific that is called as the specific plasmalogen, that is the PAF, that is the platelet activating factor. Now, plasmalogens are again the phospholipids. They also contain the choline. They also contain the ethanolamine. Likewise, but they are far little bit different. What is the difference? Now, this plasmalogen synthesis starts with the dihydroxyacetone phosphate. So whenever there is a dihydroxyacetone phosphate is present, now what happens, how the reaction does happens, we will first consider dihydroxyacetone phosphate will take up the acyl group from acyltransferase and it will form one acyl dihydroxyacetone phosphate. Now here again at C1 position by ester linkage there will be formation of one acyl derivative of dihydroxyl phosphate, and which subsequently get acted upon by another enzyme synthase to form one alkyl dihydroxyacetone phosphate. That is the significant reaction. Instead of ester linkage post generation generated, it becomes the ether linkage at carbon number 1 of the dihydroxyacetone phosphate. Now still there is a carbonyl group. Uh, Group is present, which subsequently get reduced by reductase enzyme. This reductase enzyme utilizes NADPH as a cofactor and it forms 1-alkyl glycerol 3-phosphate. Now this 1-alkyl glycerol 3-phosphate will take up the another acyl group at C2 position and it will form 1-alkyl 2-acyl glycerol 3-phosphate. Then it will subsequently form 1-alkyl 2-acyl glycerol, and again the CDP ethanolamine will be there present and there will be formation of 1-alkyl 2-acyl 3-phosphoethanolamine will be produced. That is one more intermediate, and this 1-alkyl 2-acylglycerol 3-phosphoethanolamine subsequently undergoes the desaturation reaction. Again, utilizing the NADPH as a cofactor, it requires cytochrome B5 for this reaction and it forms the plasmalogen. This plasmalogen is it contains the unsaturation 1-alkenyl 2-acylglycerol 3-phosphoethanolamine. So these are the components of the plasma membranes of different tissues, like ethanolamine containing plasmalogens are predominantly present in the myelins. Now on the other side, 1-alkyl 2-acylglycerol can take up like CDP ethanolamine, there will be CDP choline. So it will form the first 1-alkyl 2-acylglycerol 3-phosphocholine, and this product undergoes subsequent again the phosphorylation uh, phospholipase reaction, whereby rendering the remole of fatty acid at C2 position, and it is replaced by acetyl transferase by acetyl group, and it forms finally the 1-alkyl 2-acetyl glycerol 3-phosphocholine. This compound, this phospholipid is known as platelet activating factor. This platelet activating factor, as you know, you might have studied during the chemistry of phospholipids, it is responsible for the platelet aggregation and degranulation. So it is also has this platelet activating factor has got hypotensive activities, properties. It has got ulcerogenic properties. So it is responsible for increasing the airway and pulmonary edema, lungs. It can mediate the hypersensitivity reaction. It is involved in the inflammatory reactions, and it can also generate the free radicals, involved in the formation of free radicals. So that is about the platelet activating factor. And plasmalogens containing ethanolamines are predominantly found in the myelins, whereas plasmalogens containing the choline, they are found in the cardiac muscles. So in this way, these different plasmalogens synthesized. Now we'll consider about the next part about the sphingolipids. Now what is sphingolipids? Now once we have completed about the phospholipids, so phospholipids in all together mostly are glycerophospholipid, means the backbone is glycerol, three carbon compound glycerol, and these different phospholipids are involved in different vital activities, as we have seen already. Now instead of this glycerol, if the alcohol is different, that is a long chain aliphatic alcohol, which is product of combination of palmitic acid and amino acid serine, we get a long chain aliphatic amino alcohol, what is known as sphingosine. So this sphingosine is first how it is synthesized. So first palmitic acid, we have already seen the synthesis of palmitic acid, fatty acid synthesis. So this palmitic acid get activated to form palmityl coenzyme A, and it will combine with the amino acid serine, and it subsequently undergoes the decarboxylation reaction, and there will be a reduction utilizing NADPH plus H plus. This requires also pyridoxal phosphate, and finally we get the product that is a sphingosine. Now this sphingosine molecule is nothing but the long chain aliphatic amino alcohol. It contains the amino group, it contains the alcoholic group. Now this CO group carboxylic group has been given out, it has been sacrificed, and there will be formation of sphingosine. Now this sphingosine subsequently combines with the fatty acid. Now these fatty acids, predominantly mostly C24 carbon 24 fatty acids, are uh, utilized by combining with this sphingosine amino alcohol through the amide linkage. So this amino group of the sphingosine is involved in the formation of amide linkage with the long chain fatty acid, and a product form is known as a ceramide. And this ceramide is just like a phosphatidate. Phosphatidate is the common intermediate for triacylglycerol and other phospholipid synthesis. Likewise, in sphingolipids, ceramide is the common intermediate for the synthesis of sphingospholipids. The only product is sphingomyelin. It is the phosphatidylcholine and diacylglycerol. So there will be formation of sphingomyelin. Phosphatidylcholine becomes the source of choline moiety, and it will release diacylglycerol. On the other hand, there will be formation of sphingomyelin. So that is about the synthesis of sphingomyelin, and instead ceramide can get added with different carbohydrates. We get the different glycosphingolipids. Now again, we'll see this in a imaginary how. So this is the sphingosine part. That is the amino alcohol, and when it combines with long-chain fatty acid, we get the product that is known as the ceramide. And the ceramide, then there will be formation of phosphocholine. We get the sphingomyelin. We get the sphingomyelin molecule. Now, regarding the sphingomyelin, once this is formed, the excess of sphingomyelin will be catabolized by the enzyme sphingomyelinase. Now this sphingomyelinase will release the ceramide and phosphocholine. Now if this enzyme is deficient in the cells, so it leads to the, because of the enzyme sphingomyelinase, the sphingomyelinase has got the genetic deficiency, inherited deficiency, and because of this, so sphingomyelin will not be degraded, will not be hydrolyzed and lysosomal enzyme, and it will lead to the accumulation of more and more sphingomyelin where it is not catabolized. So like spleen, liver, and brain and nervous tissue, where the sphingomyelin gets accumulated, definitely there will be enlargement of spleen and liver, and then there will be mental retardation, and most of the conditions below 2 years of age, this disease is fatal. Even before the knowing of uh, what is the cause, that child can sacrifice life. So that is about the sphingomyelin and its metabolism. Now during the synthesis, so already we have seen, and there is a Newman's peak disease. The next we can have cerebroside and ganglioside. Now these are the glycosphingolipids. Now again the glycosphingolipids, backbone is the same. Sphingosine combined with the fatty acid. Now here I have shown that is C24 cerebronic acid. So, and then subsequently there will be addition of a carbohydrate, glucose or galactose. So it becomes the glycosphingolipid. Now, whenever there is a synthesis, again here the same process is there. Synthetic pathways, sphingosine will be produced. Then there will be combination with the fatty acid, amide linkage will be there, and ceramide formation will take place. Now, it requires the oxidation by FAD molecule is involved during the ceramide formation, and during the formation of sphingosine, already we have seen NADPH will be utilized, and by the CDP glucose transferase. So CDP glucose transferase or galactose transferase, we get this glucose or galactose will be incorporated in the sphingosine to form the glucosyl or galactosyl ceramides. Now there are different, these are known as the cerebrosides. cerebrosides. So there will be glucosyl cerebrosides or galactosyl cerebrosides. Now when the fatty acid is different, now here we have shown cerebronic acid. The compound is known as cerebron. Now instead of cerebronic acid, if there is a lignoceric acid is incorporated, we get the kerosene. And these are the different examples of the cerebrosides. Now when there is a nervonic acid is added, we get the nervon. If there is a oxynervonic acid is added, we get the oxynervon. So these are the different types of the glucosyl or galactosyl cerebrosides. So these are the products. Then how these are synthesized. Then the next, they can also get sulfated, and by taking the sulfate donor phosphoadenosyl pyrophosphate PAPS, which becomes, and the enzyme will be microsomal transferase enzyme, which can transfer the phosphate group to this glucose or galactose molecules. They can be sulfated, and we get the sulfated derivatives also of these cerebrosides. And subsequently, these groups can be hydrolyzed whenever the excess of these cerebrosides are synthesized. No doubt, if their function is over in the plasma membranes or in the tissues, so they can be degraded. Now, if these degradative enzymes are deficient, we get the different disease. One of them is the Gaucher's disease. It is an autosomal recessive disease whereby the enzyme beta glucose cerebrosidase is deficient, and there will be now this which cerebroside, the kerosene accumulates, and in the liver, spleen, brain and bone marrow. Ultimately it will lead to the mental retardation, poor growth, and ultimately the disease will be fatal. So that is about the cerebrosides, and the disease is known as the Gaucher's disease, whereby there will be glucocerebrosidase enzyme will be deficient. Now there is another disease that is known as the metachromatic leukodystrophy. Now here this aryl sulfatase A enzyme is deficient, so sulfate group will not be efficiently released, so that will get accumulated. 3-sulfo-galactosylceramides, they will get accumulated, and it results in the demyelination and or complete absence of myelination, and it results in the mental retardation. There is one more, that is that Krabbe's disease, whereby this beta galactosidase is deficient. So if it is a galactosyl cerebroside, then beta galactosidase enzyme will be acting to catabolize this cerebroside, and if that enzyme is deficient because of the inborn errors, so galactosyl ceramides will accumulate, and definitely as these cerebrosides are present predominantly in the brain and nervous tissue, so mental retardation, myelins will be poorly generated or are mostly absent, etc. So all these types of the symptoms, clinical pictures will be observed in this cerebroside. Part is the gangliosides. What happens in gangliosides? Gangliosides are little bit complex process, because in cerebrosides there is either glucose or galactose is added, as we know already. So glucose in cerebrosides or galactose in cerebrosides. Now instead of glucose or galactose, if there is a complex oligosaccharide is added, then we get the different gangliosides. So there will be glucose, galactose. In addition to this, there will be some amino sugars and their derivatives can be added. So if there is a glucose and galactose is added, and it's oligosaccharide chain, glucose galactose is not the only one molecule. It can get polymerized to form oligosaccharide, and that can be added, and it becomes the lactosyl. Glucose galactose from the lactose. So lactosyl ceramide, or it is also called as the globoside. Then there are other gangliosides present in the ganglion cells. So here it is the combination glucose, galactose, galnic and galnic, and acetyl galactose and neuramic acid, NNA, etc., galactose, neuramic acid, different acids. These are the complex amino sugars, just we have seen in the heteropolysaccharides. So these are added. So here it is the, if this arrangement is present, that is the GM1, ganglioside 1, then ganglioside 2, glucose, galactose, galnic, and nuic, and GM2, and then there will be a formation of GM3. So by different transferase enzymes, these groups will be added to the ceramide group, and we get the GM1, GM2, and GM3. Now again, these are catabolized by different enzymes, and if the enzyme hexosaminidase is deficient, so it becomes the basis for Tay-Sachs disease, whereby this GM2 is to be catabolized efficiently but will not be metabolized efficiently because of the deficiency of hexosaminidase, and it will start accumulating in the brain nervous tissue. So gangliosides are mostly found in the ganglion cells. So mental retardation, weakness of muscles, muscle weakness, blindness, all these will be the symptoms observed. In Tay-Sachs, there are different variants are also have been reported, like hexosaminidase A and B deficiency. Different hexosaminidase are present, and if they are deficient, the different variants can be observed. There is one more, that is the what is Fabry's disease. Alpha galactosidase is the deficient. So in this globotriaosyl ceramide, globotriaxexos ceramide accumulates, and it will lead to the screen rashes, kidney failure, and it is an X-linked recessive condition. So